Welcome, everyone. Thank you for joining us today. My name is Jenny, and I'm a technical application specialist at IVM Technology. Today's presentation will focus on the latest advancements in live animal imaging at the cellular level. Our presenter, Hayden Kwon, a senior technical application specialist at IVM, will be discussing the application possibilities of visualizing immune response dynamics in live mouse models using intravital microscopy. Our presenter, Hayden, holds a master's in life sciences and has extensive experience in both clinical and preclinical imaging and research. During the webinar, if you have any questions, please use the Q&A box below. We will also be addressing some questions submitted prior to the webinar. Hayden, the floor is yours.
Thank you for the greetings, Jenny. Hello, I'm Hayden Hyungjin Kwon, application specialist from Technical Applications Solution team here at IVM Technology. So first of all, thank you for attending today's webinar, and also it is very honor to have you all. The presentation will proceed as follows. So first, I will provide a brief overview of IVM Technology's intravital microscope, and following that, I will present in vivo imaging applications performed by IVM's technical application team regarding immune cell dynamics imaging in living mouse models. At the end of the webinar, you can see how manipulate the device for imaging inguinal lymph node with recorded video like a virtual demonstration. You may be aware of the conventional in vivo microscope, but these are actual macro scale scopes, so it was quite difficult to see the mechanisms of the disease development or drug biodistribution in live animals because you could only acquire whole body imaging data. To overcome this hurdle, IVM Technology tried to develop an optimized micro-scaled imaging system, IVM, for cellular level visualization, and now we could visualize several cellular dynamics, disease development, and drug efficacy delivery inside of the living body on real time. So, thanks to this superb in vivo imaging system, you could acquire real-time 3D imaging in various cellular activities and molecular works directly, which allows for the direct visualization and analysis of delivery and efficacy of newly designed biotherapeutics in preclinical studies. I'd like to introduce our dual mode confocal and two-photon microscope model. So this is the most compact intravital microscope in the market, ideally suiting your researches. It has various key features such as cost and space saving, easy installation, and high usability, hands-free maintenance, and all-in-one package. And in this slide, I like to emphasize the dual mode usage of confocal and two-photon laser. So we've obtained this neuromuscular junction within Thyron YFP transgenic mouse, expressing endogenous GFP in neuron. And blood vessel was labeled with anti-CD31 antibody conjugated with the fluorophore in blue. So with only confocal laser, we'd see the neuron and the blood vessel with fluorescent signal. But with the photon laser, we could have visualized the actin myosin structure in muscle with second harmonic generation signal, which is no need for labeling separately. So if your researchers need to see the accumulation of fiber, collagen, or muscular structure, the photon laser would be the perfect choice. So IVM is the world's leading company providing new all-in-one system with the best intravital imaging performance. So this all-in-one single box package enables the easy installation, operation, and maintenance. And it has also co-optimized hardware and software for superb intravital imaging performance with ultra-fast polygon scanner, temperature monitoring system, and motion compensation and AI denoising software. especially with automatic hassle-free motion compensation functions and various apparatus such as live animal maintenance system and various imaging window system for wide range intraviral applications, users easily can get the high quality in vivo data without any limitation in imaging various live organs. So this is the key technical feature one. So IBM's imaging system is equipped with ultra-fast polygon mirror scanner, speed up to 50 frames per second, leading to capture fast dynamics of cells like red RBCs or the green cells flowing inside of the blood vessels. So you can see this video as well here. So fast moving RBCs or and the fast moving immune cells inside of the blood vessels in the cardiac system. So you can easily and successfully capture all the movement of each cells. And this is the key feature too. So it's especially with animal motion compensation function, it enables an automatic hassle-free high precision motion compensated imaging by facilitating frame by frame motion compensation. It is the most optimal solution, particularly for rapid moving organs like heart and lung, as it can compensate the X, Y, and Z movement with real-time acquisition of compensated image data right away. And this is in vivo real-time video of heart imaging. So in this side-by-side comparison, even though raw imaging data has high degree of movement, but when we apply the motion compensation function, we can directly acquire the high quality of clear image without any shifting artifacts. So without motion compensation, only these blurred images can be obtained due to motion artifacts, which distorts the quantitative cellular analysis of imaging data. And please note that any other conventional standalone microscopy only can acquire these shifted images, like in the middle, from the same live mouse model. So thereby, only IVIM has optimized the in vivo solution, especially only for intravital heart imaging and the lung imaging in these research fields. To maximize the utility of the video rate imaging capability of IVIM's microscope, we developed an AI model to efficiently denoise any real-time videos obtained by our microscope. The outcome preserved the high temporal resolution of the video while removing noise. And surely, during the denoising procedure, even the small molecules like nanoparticles, you can see as in red, are also successfully imaged. So it was very difficult before because previous denoising AI models lose temporal resolution with denoising. So this work was published in Nature Method last year, and IBM has an exclusive license for the global market. Also, IBM further developed more advanced internally trained AI model to speed up the denoising processing more than 50 times. And I'd like to share the last key feature of the IVIM technology. So especially for longitudinal imaging, we are providing in vivo imaging window chambers that are fully optimized for organ-specific applications. And we could, through this apparatus, we could successfully obtain the heart and the lung imaging data using the specialized imaging chamber with tissue motion stabilizer to hold the tissue very still to reduce the artifacts induced from heartbeats and breathing. And in the case of imaging the liver, kidney, and other abdominal organs, we could implant this abdominal imaging window on the abdomen, which makes us acquire long-term imaging data up to almost 2 months. Thanks to the advantages of being able to image organs in a living animal's body at the cellular level, IVIM Technology's current biomicroscope is used for imaging various disease models, including cancer models, dementia models, and sepsis models, including immune cells in lymph node or other organs. And also you can see the interactions of the therapeutic molecules in various animal disease models. Okay, for now, I will share the imaging data for observing the wound healing and immune cell motility in living mice. In this study, we employed lysine GFP transgenic mice, which exhibit endogenous GFP expression, specifically in neutrophils and macrophages. Additionally, we utilized an in vivo labeling kit consisting of anti-CD31 antibodies conjugated with the far red fluorescent dye to specifically stain blood vessels. As shown in this video on the right, we successfully stained the blood vessels. So in the timeless movie captured at 30-second intervals, you can observe the active movement of the immune cells. The images in the upper panel represent the ear skin of the control group, which was not exposed to the radiation, while the images in the lower panel shows the test group after radiation exposure. So given that both images are at the same scale, we observed approximately 6 times more neutrophils gathering at the radiation exposed site compared to the control group. Additionally, since the temperature difference between the non-exposed and exposed areas was only 0.2 degrees Celsius, we further confirmed that this was an acute immune response triggered by radiation, not a result of temperature changes. And again, most macrophages did not move around, so their motility changes are well correlated with the change in neutrophil and macrophage count before after irradiation. In this second skin imaging experiment, we used CX3CR1 GFP transgenic mice, which express endogenous GFP in monocytes and macrophages to visualize the immune responses in ear skin. So a wound was induced within a 31-gauge needle puncture, and the KRS inhibitor, known for disrupting cell-to-cell adhesion, was applied 4 times. Imaging immediately after the wound, as well as 1 and 2 days later, revealed a significant reduction in immune cell recruitment around the wound site in the KRS inhibitor-treated test group compared to the PBS-treated control group. So this reduction was attributed to decreased cell-to-cell adhesion and migration in the test group. So we can see in this slide, you can see the analyzed process data in bar graph. So you can compare the actual number of the recruited immune cells with or without treating KRAS inhibitor. So next, I will share the result imaging data for tracking immune cell motilities in mouse lymph node. Lymph node is a small organ of immune system acting as filter or transfer foreign particles. The lymph nodes are distributed through the whole body and connected to lymphatic vessels. Although lymph nodes are far away from each other, they function as a single entity mainly because of its principal cellular constituent, lymphocytes. The lymphocytes are intrinsically mobile and continuously recirculate in large numbers between lymph nodes via the blood and the lymphatic vessels. This data showcases various immune cell movement within the popliteal lymph node. Additionally, in the previous slide, we could observe the movement of the B cells and the T cells. And similar to T cells, B cells were also visualized passing through the fibroblastic reticular cell, RFC network of the lymph node and exiting outwards. In the lymph node, there are specialized blood vessels called high endothelial venule, or briefly called HEV. You can easily find HEV in lymph node because of their large size and the thick vessel wall. HEV is main entrance into the lymph node for circulating lymphocytes. In normal condition, HEV allows only lymphocytes to enter from blood. For last decade, multiple steps of T lymphocyte entrance in HEV and related oxygen molecules have been identified. However, the exact behavior of interaction between rapidly flowing T lymphocytes and endothelial cells remains to be analyzed. In addition, the transmigration of T lymphocytes across endothelial cells has not been cleared because of the lack of proper in vivo endothelial staining methods. However, with our IVIM technology's real-time in vivo imaging capability, we were able to observe the migration of cells passing through the HEV. So in this study, we stained fibroblast reticular cells which compose the microarchitecture of lymph node using elixaflor 647 conjugated anti-ERTR7 antibody displayed in blue. Additionally, we injected an in vivo labeling kit composed of anti-CD31 antibody to stain blood vessels and CD3 antibody to stain T cells, resulting in red and green signals for each cell types, respectively, before proceeding the imaging. In this video on the right, you can observe the T cells flowing inside of the lumen of vascular endothelial cells, then adhering to the vascular structure wall, and subsequently migrating towards the FRC site, which is the other site. As you can see in the video on the right, we were able to visualize in real time the process where T cells flowing within the blood vessels in the lymph node adhere to the vessel wall, pass through, emerge, and all within just a few minutes. This data reveals that not only T cells but also B cells utilize the perivascular channels within the lymph node to traverse from the HEV lumen to the blood vessel, and even though the FRC network. So remarkably, we found hotspots for the trans-EC and trans-FRC migration of T and B cells. So interestingly, T and B cells preferentially share the trans-FRC migration hotspots, but not trans-EC migration hotspots. Furthermore, trans-FRC migration of T and B cells preferentially occurred at FRCs covered by CD11C positive dendritic cells in HEVs. So these results suggest that HEV ECs and FRCs with perivascular dendritic cells delicately regulate T and B cell entry into peripheral lymph nodes. So with IVIM's intravital microscope, you can get high-quality live cell imaging in almost all internal organs, not only lymph nodes, but also including brain and thyroid, heart, or the kidney as well. So far, I've shown you imaging data that we've performed before. So now let's move on to the virtual demonstration session to demonstrate the actual imaging process for lymph node visualization. And so this is the brief scheme for today's lymph node imaging. I anesthetized C57B6 wild-type mouse and then removed the hair on the right side of the body. After exposing the inguinal lymph node on the right side by simple surgery, we conducted lymph node imaging using anti-CD31 antibody conjugated with the fluorophore to stain blood vessels in red. And also the mouse was injected with small particles in green to see the particle flow inside of the vessel. Oh, okay. Oh, sorry. Okay. So I'm currently showing the actual appearance of the equipment. So at the top, you can see the main body of the microscope, while at the bottom there are various supplies. Zooming in the bottom, you will notice three main devices. The main power supply for the microscope. Okay, so the main power supply for the microscope, and each power supplies for the two photon and the confocal laser each. And next, I will explain the main body in more detail. So, okay. If we go up to see the main body, there is a stage where the animal model is placed for imaging. And on the ceiling, there is a turret that can hold up to 5 optic lenses. And next to it, there are the 5 ports where you can connect up to 5 temperature sensors. So for example, if you want to keep the stage warm to maintain the mouse's body temperature stable, you just need to connect this cable to the port. Once connected, the animal maintenance system automatically measures the temperature and adjusts it to the set temperature to maintain the mouse's body temperature. You can also use the rectal probe to measure the core body temperature of the mouse, and with just one click, you could adjust it as necessary. So using the jog dial at the top, you can move the stage along the X, Y, and Z axis to proceed with imaging. And now I will show you the operating mouse in greater detail. So I prepared for this mouse for imaging the right inguinal lymph node of the mouse by anesthetizing it, shaving the area, and then making an incision to expose the lymph node. Afterward, I replaced the cover glass over the tissue to prevent it from drying and to match the refractive index required by the lens. And now I inserted the stage into the chamber, close the door to start imaging. Okay, now I will show, here we have the IVM engine featuring a user-friendly interface. So on the main screen, you notice the separated displays for the TAPI, GFP, RFP, and the Sci-Fi channels each. And additionally, there are displays for the integrated live video main displays and a window for captured images. So to ensure the optimal excitation and detection during in vivo imaging, I've already adjusted the laser power and detector values. So by activating the scanner and shutter, we can locate the focal plane until the signals are displayed, enabling us to observe the morphology of the lymph node blood vessels. So using the jog dial, we can navigate to find the best field of view on the cover glass. So keep an eye out for large blood vessels with multiple branches and vasculature, as they are ideal areas for capture the signals. So in this experiment, the red signals represent blood vessels labeled with anti-CD31 antibody conjugated with the fluorescent dyes from the Sci-5 channel. And the green signals represent fluorescent particles. So we can observe the green labeled particles in action as they perhaps flow in the endothelial surface or firmly adhere to it with a sense of purpose. So with each observation, we inch closer to unlocking the secrets of vascular endothelial dynamics and the delicate balance between drug response and circulatory functions. So for this imaging session, I used the Z-stack imaging modes, Z-stack and mosaic imaging mode. So this approach allows us to obtain the comprehensive view of the vasculature across its entire depth. So After finding the right focal plane with using the jog dial, I loaded the z-coordinate to capture the images starting from the top to the bottom with a 10 micrometer interval. So this imaging depth will be at 60 micrometers. Once the images are captured, we can use IVM Studio to post-processing software to stack them together. So you can use all the functions from ZStack to time-lapse in combination, but to be respectful of your time, I use both ZStack and Mosaic at the same time. So move the stage to find a suitable focal plane and to visualize the cells and the microbits in the lymph nodes. So to get the large field of view of imaging data of the target organ in case of like lymph nodes, we could utilize the mosaic function here. So just find the right spot and set the start and final coordinates, and the software will calculate the total image number. So for now, we will get a 2 by 2, total 4 images, and stage will move automatically during imaging once you've done the settings. So there's nothing to do for you. And during the imaging session, I'd like to share the imaging data that was captured through this imaging session. So the experimental conditions remain the same. So on the left side of the screen, you can see the lymph node imaging taken with the 25x lens, providing the high resolution view. And at this spot, I recorded the video so we could get the information about the movement of the particles in the live mouse. So on the right side, there is a video extracted from the 3D build data, which is not shown right now. Maybe you can see in shortly. So you can see the 3D build data allowing us to visualize the vasculature of the lymph node from different angles. And I extracted the orthogonal video from the surface to the bottom to look into the vessels and particles more precisely. So if the video is over, we can move on to the post-processing part. Yeah, I would start to demonstrate how to post-process the acquired data using IVM Studio, our software for 2D, 3D, and 4D in vivo data processing. So please note that due to the time constraints, we may not be able to cover every function in this short webinar. By adding the highest directory folder into IVM Studio, you will have access to all the categorized files. So within the software, you can adjust and improve the quality and visibility of the images, such as modifying the color balance, and brightness, and also the contrast of each individual images. And once the necessary adjustment has been made, we can play the set of ZStack images with the ZStack stamp and the scale bar inserted for better visualization and analysis. So this allows you to observe the acquired images in a sequential manner, gaining insights into the lymph node structure across the different focal planes. And it is completely possible to perform basic functions such as making Z projection and furthermore, quite time-consuming and demanding one like making mosaic, also called the stitching. And additionally, we can utilize the depth-wise acquired datasets for 3D reconstruction. by making a few adjustments within the software, by adjusting the color level, we can generate the video clip that provides the horizontally rotating 3D view of the lymph node structure. And this visualization method complements the 2D images and offers a comprehensive understanding of the tissue's organization and architecture. So I'm gonna stop the video to move on to the next part. Okay. Throughout this webinar, we have explored the process of in vivo imaging using the intravitreal microscope, along with the associated hardware and the software. So thank you for paying attention, and if you have any questions, please feel free to ask through the Q&A box.
Thank you very much, Hayden, for the great presentation. We'll now take some time to answer questions. The first question is, can we have the PowerPoint after the training?
Sure. If you can send the email from on your side, we will send you the PowerPoint slides for you. So thank you for having interest in our slides.
Thank you. And the next question is for your CD31 antibody staining of the ear, which far red fluorophore did you use? What was the excitation wavelength? What filter set did you use to detect anti-CD31 signals?
For visualizing the blood vessels in the ear imaging study, we used the CD31 antibodies conjugated with the fluorophore, like Alexa-Fluor 647 or FSD 647. So this fluorophore has excitation and emission at each 640 and over 640. So I could send you the filter set after this webinar is over. We have optimized filter sets for visualizing the various cells or other molecules inside of the living body with several fluorophore. So maybe you can use the filter set for your research.
And the next question is, for lymph node imaging, what objective did you use?
We can use up to 5 optic lenses in one intravitreal imaging system. But in this study, we recommend to use 55 water emulsion optic lens to have the high quality imaging data. But you can use any magnification lens for your study. So we have optimized for using several optic lenses with our system. So you don't have to worry about that.
Thank you. And the next question is, do you have an in-house software to analyze T and B cell entry in the lymph node, or did you use software from other vendors?
Okay, for analyzing the T and B cell entry in the lymph node, we use a third-party software like Imaris or something else. But we are trying to optimize to calculate the velocity or the velocity for basic analysis like the trafficking or the measuring the velocity or the cell numbers.
And the next question is, there is applications in bone repair, wound imaging by IVM.
Yes, actually, several months ago, we have one published paper which is regarding the osteoclast imaging with 2-photon microscope within the mouse bone. So, if you want to get the information about the paper, we can send you the theme and the name of the paper for you.
And the next question is, will it be possible to also use this microscope for organoids or organotypic slice imaging with a thermostatic chamber?
Yes, we have experience in visualizing the organoids in the liver after inducing the injury in the liver. So maybe, I don't know the exact type of the organoids you're interested in, but I think we could successfully visualize the organoids in the living body.
And the next question is, do you have local tech support for US customers?
Yes, we have distributors in the US market. So maybe if you're interested in purchasing or using our services or something else, we could send you the information of our distributors via email.
And we'll take one last question. How are bone marrow images acquired?
Okay. Bone marrow imaging is one of the easiest part for visualizing in vivo. So because mouse has its own bone marrow system on the skull. So after incising the scalp with just simple surgery like opening the scalp, you can see the bone marrow very easily after mounting the mouse on the stereotactic stage. So maybe if you use the specific or suitable cells, molecules, or something else like for your study, we can see your target molecules in the bone marrow flowing inside of the marrow or in the blood vessels in the marrow.
So it looks like we've covered all the bases, and the webinar will be available on demand on our YouTube channel at Iving Technology. We offer both hands-on and virtual demos, along with free imaging test experiments using your samples and training for animal surgery models. We thank you all for having so much interest in us, and due to time, we weren't able to answer all your questions. But if you have any more questions for us, feel free to reach out to us at information@ibeamtech.com. Thank you once again for joining us today, and we look forward to seeing you next time. Thank you.